Thank you, Leon. I will share my screen now. Okay, so it should be visible by now. Okay, full screen, great. Perfect, thanks, Leon. Okay, so hello, everyone. My name is Michael Schirner. I'm a researcher in the brain simulation section of the Charité Universitätsmedizin Berlin and Berlin Institute of Health. Just short my background, I studied bioinformatics and applied computer science and work in, basically in brain simulation mostly. So I'm simulating brains and I'm trying to create brain network models that explain human behavior. And I'm also involved in setting up cloud infrastructures where researchers can process their data online, collaboratively, with high performance computing resources in the background. And we will also talk today about these cloud infrastructures, but the main topic is MRI processing for the virtual brain. And this session is basically the follow-up on the awesome talks that Leon Martin gave about MRI processing. So I think one could say that the talks by Leon Martin gave the theoretical background, and these are now a little bit more on the practical side, I would say. So the practical problems that show up when one tries to do MRI processing. Okay, so what is the goal actually? Why do we want to process MRI data? So the goal is to construct brain network models. So brain network models are an abstract representation of the brain, and you already had a lot of information about those in the previous lecture, so we will just recapitulate this information. So the actual goal is that we want to understand and create explanatory theories, so explanations why brain activity is as it is, why brains operate and function the way they do, basically. And because brains are very complex and brain data is very complex, also the underlying theory or the theory that explains these data are usually or often very complicated, and it's hard to think about these theories and to deduct new insights from these theories. Therefore, we have models, and we have mathematical models, sorry, that are then turned into computer models that are then simulated on computers. And with this, we can basically bridge the gap between data and theory. So in our case, the models simulate mostly the large scale. So the goal here is that a whole brain can be simulated. And therefore, we usually don't simulate on the level of individual neurons, but on the level of neural populations. And a typical motif for simulating a brain area is a coupled excitatory and inhibitory population. So we have an excitatory population that is coupled with an inhibitory population, and they are both also recursively coupled and excite or inhibit themselves and each other. And this short video animation now gives basically an overview what data we can really extract from MRI data. This is all you can see is extracted from MRI data. It's all from a living human being, and all of this can be really reconstructed from the MRI data on this resolution. So this is not faked or anything. This can all be constructed by doing MRI processing. We have surfaces of the scalp, of the cortex, of the skull. We can track the white matter fiber bundles that connect brain areas and then parcellate the brain into a set of different regions according to a certain atlas, and then we can aggregate this complicated information and make a simpler model out of it. It's still a complex model, but it's simpler than the very high dimensional data that one can get out of MRI, for example. So what types of data are used to inform brain models? So the three most important modalities in order to construct a brain network model is T1 weighted MRI, which gives a nice contrast for anatomical structures. So one can differentiate gray matter and white matter, for example, and or for example, like you can see here, we can see a lot of structures very nicely, even with a blank eye. So the T1 weighted images are used in order to basically localize function and to know where we are in the brain, basically. But the more relevant information for the brain network model is the diffusion weighted MRI, actually, because this gives us the connectivity between brain regions. So these are the white matter fiber tracts that connect different brain areas. And this is important to inform the brain network model, because this is one structural ingredient of the model, the connection strengths between brain areas. And then fMRI is very popular and very exciting technique because it basically lets one monitor brain activity on a very high resolution. So, these voxels, these volume elements of this data set can be as small as 1 to 2 millimeters. And with this resolution, we can monitor brain activity. And from fMRI, we usually extract functional connectivity, the network patterns, and use these to then again the network model. So what types of connectivity do we use? So as we saw earlier, we divide the brain into a set of different regions, and we reconstruct these white matter fiber bundles that connect the different regions. And then we basically count them more or less to arrive at this information, the weights matrix. So for each pair of regions, we can see how strongly are they connected. And we can also reconstruct the tract lengths. So we can just measure how long such a reconstructed fiber bundle is, and this gives us its length. And from that, we can then compute a time delay. So the length divided by a conduction velocity, like for example, brain signals would usually travel with a velocity of about 10 to 100 meter per second. And we can basically then compute the time delay of interaction. And this is how a typical SC matrix looks like. So one obvious thing that we see is that within one hemisphere, so here what we see, the first 100, about 80 regions are all the left hemisphere, and then the next 180 regions are the right hemisphere, and then come subcortical structures. And we can see here there's a strong connectivity between the same hemisphere and a relatively low connectivity between the homologue on the other hemisphere. And when we look at the distribution of connection weights, so when we look at the distribution of weights in this matrix at the histogram, we see that the magnitude, the scale of these values stretches very large, and the majority of connection weights is actually very, very small, and only a very small number of connections is actually very strong. This may result from biases that happen during tractography, but we will come to that. So another type of connectivity we will not so much talk about here is short range, large scale connectivity. So long range, large scale connectivity, which we just had, is long range because it's these long white matter fiber bundles. And the short range connectivity are basically the short lateral axons that connect neighboring populations on the cortical surface. And here the connection strength is even harder to measure, and therefore these generic connectivity kernels are used where the connection strength falls off with distance. Or for example, it could also be inhibitory at a certain range, which is some typical cortical motive, and then only later becomes excitatory again. So to now integrate this MRI data and what we extract from it with brain network modeling, it's best to look at the actual equation. Basically, there we see where all of this actually comes in, and basically every brain network model can be expressed with this generic. Sorry, I don't find my cursor anymore. Yeah, there. The go-to window is in the slide. I don't get it out of the way. Oh. Okay, so I'll try it anyway. So the basically on the left-hand side of the equation is a differential equation. So this means basically we can compute the time evolution of this equation. So this is an equation that is integrated over time. So it's xi depending on the time t over dt, and we can then integrate this, a differential equation. And the most important ingredient, or the one important ingredient, is the neural mass model. So this is the local population, basically the local population. So this can be our excitatory and inhibitory equations, basically that simulate how synaptic currents arrive at this population, how they are converted to firing rates, and how they are then again converted to synaptic activity, for example, and sent to other populations. And this function then takes the current state of the population and computes the next state of the population. And the next state of the population depends on the current state, as we saw, and on the sum of all its inputs. So this is basically the first term on the right-hand side is what the local populations do, and everything that comes after this first term is basically inputs that go into this population. So the second term here is the input that comes from the long-range large-scale connectivity. And here we have the structural connectivity matrix. And basically all we do is we just produce a weighted sum of the activity of the connected populations. So we look at all other populations. What is the activity? We weight it according to the connectome, to the structural connectivity, and sum it up. And we do this in a time delayed manner. So we only sum up the activity after the appropriate delay that was needed for the information to reach this population. And then one important parameter is usually this global coupling scaling factor that rescales the whole structural connectivity matrix such that it's appropriate, it gets absolute values, because it's only relative values what you get out of diffusion weighted MRI. We don't get like an absolute current in ampere, for example, but we fit this then. And then this second term here, sorry, the third term here is the same like the second term, but for this short range, large scale connectivity. And then here we have injected input. So we can also inject all kinds of inputs. So for example, we did a study where we measured simultaneously fMRI and EEG. So the people were lying in the scanner and they had EEG caps on their heads. And then we used this EEG and we injected it into this model via this term. And the model then produced the fMRI activity that we measured while the subjects were lying in the scanner, which is pretty cool because it connects these different modalities, basically. And then the last term is usually noise. So if we don't have anything that drives the model, then we usually need some kind of afferent input, some noise that drives the populations, because otherwise they would go into a fixed point, which you maybe remember from dynamical systems lecture. And this is very similar, like it's in a real brain, right? Our actual brains are also driven by afferents. So this is more or less the simulation of this. So now we can refine our goal of brain modeling. So we have a brain model, this is this equation, and this brain model produces simulated neural activity on a computer. And then we compare this simulated activity with empirical neural activity. and we will usually then find discrepancies. And this discrepancies then allow us to find errors in the model, basically, and to explain why the model doesn't do what it should do, basically. So by comparing the simulated activity with the actual activity, we can find out what is wrong with the model, what is wrong with our understanding of this phenomenon. And this allows us then to optimize the model, to change its structure or its parameter values, such that the model produces better predictions or can better explain the observations, basically, or used to explain the observations. So it's basically MRI in, MRI out. So we put structural MRI and diffusion weighted MRI into the brain model, and what we get out of the brain model is functional MRI, fMRI. And this we can then again use to optimize the brain model by changing its structure or changing its parameters. And in addition, we can use much more or other kinds of modalities. Like, for example, Leon, which just gave the introduction. He has a paper with other authors where they used PET data to constrain the brain model. So they set parameters based on the subject's PET data. Or one can also use, for example, the heart activity. Or, for example, a questionnaire. So what I'm currently doing is I'm comparing the output of a simulation with an IQ test, basically an intelligence test. And this can also be done and can also be used to construct or to compare brain network model simulations and simulation results. And the models, they not only produce functional MRI. So these models are really biophysical oriented models. So the idea is really we simulate the biophysics of the brain, really neurons and neural populations, basically. This is the idea. So it's not an abstract model that looks for abstract features, but the simulated activity is really very similar to what you measure when you stick an electrode into a neuron, or when you have an electrode that is nearby a population of neurons, like a local field potential, for example. So we really simulate synaptic activity, we really simulate the firing of neurons, for example. So based on this true or this modeled biophysical activity, we can basically then compute all kinds of activity out of that. So this can be used to compute fMRI, EEG, MEG, spiking activity, multi-unit activity, and so on. So all of these modalities are basically there. So let's just have a little bit of a closer look to the model, and then I promise there will be no further equations. But I think it helps if we can associate with the mathematical ideas. So again, this is now a very specific model, and the equation you saw previously was like a generic form of this model, and now we have a very specific incarnation of this equation. What we can see here, we have basically now it also divided into exhalatory and inhibitory populations. And for these populations, what we do is first, all we do is we sum the input currents. So we really look what are the connection strengths that we got out of our diffusion weighted MRI data, and sum this up. And this sum of input currents, which then give an actual current in nanoampere, can then be converted into a firing rate, assuming such a sigmoidal conversion function that converts currents to firing rates. And then now we have a rate in hertz, and this again can then be used to simulate synaptic activity. So obviously, if the population is firing at 20 hertz, it will produce a higher higher synaptic activity than if the population just fires at 3 hertz. So all of these are in the end monotonic functions of each other, basically. And with this, we can basically compute also fMRI activity. So what we have now is we have simulated synaptic activity, simulated neural activity, and this neural activity which is electrochemical activity. So this is the primary activity of neurons. There is electricity and chemicals. This is what neurons do. And what fMRI measures is hemodynamic activity. So fMRI is actually the oxygenation of blood, how much oxygen is in the blood. Because when a brain cell starts to fire, or a population starts to fire, then a typical response that we can see in the nearby blood vessels is something like this. So after about 5 to 6 seconds, there will be a stark increase in amplitude, and the vessels constrict and draw fresh blood with new oxygen, basically, and sugar. And this is a very simple way of expressing the relationship between electrochemical and hemodynamic brain activity. There's also a more complicated model called the Balloon-Windkessel model. In the end, what comes out is basically a low-pass filtered version of the neural activity. So hemodynamic activity is basically a low-pass filtered version of electrochemical activity. So the low frequency components, only the very slow activity. So as we can see here, this goes over 30 seconds. So these are very slow activity oscillations, basically, that are described here. But the nice thing about fMRI, it's slow. So this hemodynamic response is slow. But the cool thing is that the spatial resolution is very high. So although the temporal resolution is quite low, we can get a really nice picture about how strongly these brain regions interact, or at least, for example, how synchronous their activity is. Depending on basically the correlation coefficient between two brain areas, we can compute such a matrix where this is a functional connectivity matrix. So every single entry in this matrix basically is the correlation coefficient between the two fMRI signals. And in this case, we have 379 by 379 data points. And this already gives a very detailed description of the network architecture of a brain, and can therefore be nicely used to fit a brain model. It's often a bit confusing when we talk about these different forms of connectivity, because now I just explained functional connectivity. So this functional connectivity is really just a statistical dependence, like the correlation coefficient between two fMRI signals. But the structural connectivity is like a static thing. So structural connectivity is this axon bundles that we use as input to the brain model. But the output then is functional connectivity. So this is the input structural connectivity, output is functional connectivity. But interestingly, it was found that functional connectivity changes all the time. So these brain networks, they are not static, but they are fluctuating all the time. And this is actually a more interesting target for fitting brain network models. It's the short-term FC. So the patterns, and these are all often more subject-specific and often also reveal more about disease, for example. And therefore, many are going into the direction of dynamic functional connectivity. And effective connectivity is basically, it gives a directional effect. So when we compute the functional connectivity between two brain areas, we only know, so this is a univariate measure. We don't know, is one brain area more active in one direction, stimulating more the other, or vice versa. And effective connectivity is a more direct measure. It gives also the directional effect. Okay, and but another representation of functional networks that probably more people know are these independent components. So ICA, independent component analysis, is also a way to reconstruct these functional networks from MRI data, and it's a nice technique because with it these networks were found. So this is a different technique because ICA in the end allows to divide a high-dimensional data set into a weighted sum of linear signals, basically. So here we have our fMRI data set. So this is basically the whole brain, all the voxels going over time. And this can then be decomposed into a spatial pattern and a temporal pattern. Sorry, this is the spatial pattern and a temporal pattern. So this is the full data set, and here is the decomposition into a temporal pattern. So here's a strong oscillating activity and a spatial pattern. So this strong oscillating activity happens predominantly in this central voxels here. And here this is different. So ICA is really a cool technique because it's very helpful for MRI processing in general. Because these resting state networks, they are really very robust in all animals. So you find them in all higher animals, basically. You find them in all humans, obviously. You find them everywhere. But it's hard to get this nice structure out. So often a group ICA is a good choice in order for pre-processing, because then one can first extract a very strong signal that only emerges when you average over a group, and then later you can then detect this signal again in the individual. It's probably hard to understand what I mean, but just keep in mind that spatial independent component analysis is a great way to separate a complicated signal into its Gaussian mixture. So all these resulting signals are then very Gaussian and gives basically a finer description of the brain area, a much more understandable description of brain activity than when we just look at fMRI data set. And another advantage is also that it can be used for cleaning the fMRI data. So for example, what we can see here, so this is one IC now, and one independent component. And when we look at its time course, we see that this independent component has some sort of drift. It continuously increases. So there's this slow drifting signal there. And this is very untypical for brain activity. So our brain activity doesn't drift over such a long time upwards. It usually oscillates much quicker back to a steady state. Such drift is more an indication of, for example, that the subject moves in the scanner and gradually moves somewhere else, basically. And this can be nicely seen with such a decomposition with an ISNA, but it's hard to see in the raw data set. But in contrast here, this is an IC that looks much more plausible. So here we have the typical frequency spectrum below 0.1 hertz, and this is a wave that looks very brainy, very fMRI network-like. So we have a slow oscillation. We have a relatively smooth areal boundary. So this looks much more plausible and would probably be retained, while the other IC would be deleted. And this is a nice way to relatively non-invasively clean MRI data. Let's have a closer look now at these different techniques and at the MRI images themselves. So in the T1 weighted MRI image, we can differentiate gray matter. We can nicely see it here. There is this gray strip. This is the cortex, basically. The cortical surface is really like a surface. It's really like a sheet. It wraps around the white matter here. And the white matter are basically these axon bundles. And here, these very dark areas are cerebrospinal fluid and mostly uninteresting for us. Then what else can we see? In diffusion weighted MRI, we can measure the diffusion of water molecules. And this is basically the signal or the information we want to extract from the data set. So if we imagine water diffusion in a glass where there are no boundaries, it's isotropic, identical in every direction. Same in gray matter. There are no big boundaries for the water, but along axons, there are myelin sheets which contain fat, and they block the diffusion of water, and therefore the water now more diffuses along the direction of these axons. And this is what we measure here with diffusion weighted MRI. So basically for every single voxel in the white matter, we can reconstruct what is the primary direction of diffusion in this voxel. So here is the corpus callosum, the large structure that connects the left and the right hemisphere in the middle of the brain. And here we can see, and this is very plausible, that there are these horizontal diffusion patterns. So here we have strong axon bundles that run from left to right, and this makes a lot of sense. And then they point upwards, and this is reflected here. Here basically we see the major diffusion direction, and tractography, or the way in order to reconstruct structural connectivity, in order to reconstruct the structural brain network, is basically we start at one region, and then all we do is we follow the diffusion direction until we arrive at another region. And this is the way to reconstruct structural connectivity, but it's far from so it comes with many problems. Just a quick overview over the modalities. So as we mentioned, we have PET, we have EEG that we can also use, but we can also use tract tracing. Tract tracing is now a more direct way to probe the structure of neural networks, because here one really injects a virus directly into neurons or neuron tracts, and then this virus affects really in the direction in which the neuron grows, more and more of this neuron. And with this, one can basically better reconstruct, or more directly reconstruct, the connection pathways. But obviously, this cannot be done on a large-scale basis. So this is used in order to validate what we get out of diffusion weighted MRI. And histology is like looking at slices of brains under a microscope and coloring these slices with a certain dye. And this can also be used to inform brain models. So there are studies that really perform slice up brains with micrometer resolution, and then a certain dye colors, for example, the cell body, or only specifically inhibitory cells, or only specifically whatever the dye targets. And with this, one can do very detailed analysis of the brain structure. And then what is the output of these neuroimaging pipelines and this MRI processing? So as we'll notice, many things repeat here. So the idea here is that we show many things from different perspectives and from a practical viewpoint to get a understanding of what we're dealing with in the practice mostly. So what we are getting from a neuroimaging pipeline is functional connectivity from the fMRI, then structural connectivity from the diffusion weighted MRI. Then we have projection matrices. So projection matrices allow us to predict the EEG or the MEG signal from the neural activity. Like I mentioned before, we simulate neural activity, and then from this neural activity, we can predict all kinds of other activities like fMRI, but also EEG and MEG. And for this, projection matrices basically describe how the brain activity, the electromagnetic field, traverses through these skull and scalp surfaces, basically, and what arrives at the sensor in the end, basically, depending on how thick different volumes are here. And region-wise fMRI, obviously, and brain maps. So these parcellations, these atlases, show us a division of a specific brain into a region such that we can find the same region in different people, in different subjects. And these brain maps are used for that. And this is really quite amazing, actually, what one can do with MRI and these pipelines. Because not long ago, all of this still looked like this. So not long ago, all of this was just hand-drawn pictures. And suddenly, with the invention of MRI and all these computing techniques, we are now at this, where we really have high-resolution reconstructions of cortical surfaces and individual structures, which is really amazing. And the Human Connectome project, so the researchers that actually have made these hand-drawn pictures in the past, David Van Essen, for example, they now also produce very high quality research about how to optimally process MRI data. And so I'm usually, when I'm processing MRI data, I try to follow their pipelines as closely as possible. Usually. So they not only defined a set of best practices for processing MRI data, but they also came up with a parcellation of the human brain that is, yeah, so plausible and so good that, yeah, for me, it's the it's really a great piece of work. And so in the next minutes, I'm basically explaining what they are doing. And not only, so in addition to all of this, they also publishing studies with really many subjects. So they have, for example, a study with 1,000 subjects, with a full MRI data set, with intelligence tests, with hundreds of questionnaires that the subjects did, basically, or hundreds of metrics at least that were taken from the subjects. And this is really a wealth of data and information that is publicly and shared for free with a very high quality. And basically, in order to process MRI data for TVB or for all other applications, one usually combines a series of low-level tasks. So one thing that one always has to do is artifact and distortion correction. So there are always artifacts and distortions that need to be corrected. Then we'll look at some artifacts in a minute. Then surface reconstruction. We have seen this. The surface is very important to know because it helps, for example, to register different brains onto each other and to segment these brains and parcellate these brains to find the same region in different individuals. This is a big goal. Then warping and alignment of standard space would be another way. So instead of finding the right region within one subject, we warp and we align and we change the brain structure of the subject such that it fits a standard brain layout, basically. And I would argue, and in the following also with some slides, that this is probably not optimal. So this is highly debated, basically, and there are better techniques now. And then signal aggregation is like a last step. After we corrected everything, we know where we are, then we usually downsample or aggregate the signals from the fMRI. So an fMRI data set has maybe 100,000 or 300,000 voxels. And then after this aggregation, so a first round of aggregation brings it down to around 90,000 data points. And then the brain atlases and the brain parcellations we usually use have around 400 regions then. So the techniques we just talked about are relatively uncritical, but some techniques that are often used remove substantial amounts of information. So for example, smoothing means that you just average the signal over nearby voxels. This is very often done to better compare data sets and to get them in a state that is more easy to use and also to increase the signal to noise ratio. But it also, for example, it leads to leakage of information from it. So if we assume that our brain region of interest is here, then if we just smooth, then information from nearby brain regions would just be averaged into our brain region of interest. And this is actually not a good thing. Temporal filtering we don't need to temporarily filter at this point. So we can later temporarily filter, but not for pre-processing. Nuisance regression. For example, people look at the motion time course. So how did the person in the scanner move? And then they would fit a model to the fMRI time course and subtract this movement pattern from the fMRI time course, because many people say basically, yeah, this fMRI time course is just movement, basically. So this is an artifact, and we need to clean it out. But this is a very invasive processing step that really may delete the actual information that you're looking for in your study. The same goes for sensoring and scrubbing. So sensoring and scrubbing, these are techniques that are really done. So there are studies that do this, where basically individual slices, time points, are just removed, because it was found that these are there are too many artifacts, for example, too much movement, and so sometimes people just delete portions of an fMRI data set, and this, of course, totally destroys the integrity of the data, and it's what you can do with it. So this must be done really with caution. Sometimes it's not done with caution, and this then really affects the interpretability of the results. So here's an example, an exemplary sequence of processing steps. So we start with an initial distortion correction, some scanner artifacts, for example. Then we make some initial alignment to standard orientation. So we look roughly where does the subject, how is the brain oriented, and bring the rough orientation, anterior, posterior, into a rough alignment, such that the following algorithms like brain extraction have it easier, because they already know roughly where they have to look. And then brain extraction then is a good technique because it helps registration. So if there's still the skull outside, this can then be problematic if we register, for example, T1 and T2 weighted images, because the skull would not be visible in T2 weighted images, and then disrupt, in the worst case, the registration. Then we perform further distortion corrections. Maybe we perform some non-linear volume registration, to MNI. So for some structures, it's also fine to perform such non-linear registration. We'll come to that in a minute. What's the problem with non-linear registrations? Then cortical surfaces are important for many analyzers. So we generate them, we reconstruct them, we segment the subcortical structures. We run free surface recon all. So free surface recon all is like the Swiss Army knife of MRI processing. It's very often used because it does a set of standard processing techniques very robustly. So usually free surfer works, and usually they don't come out spurious results. So what is important here is that all of these algorithms, MRI processing, is based on heuristics, basically. So there's no artificial intelligence that really can understand what is happening here. These algorithms basically are all based on rules of thumb, one can say, basically, heuristics that look for certain things that may or may not be there, basically. Then folding-based surface registration is good to get this complicated cortical surface a bit under control and to learn where we are. And then a good step is these myelin maps, because they really allow for a good functional localization. And also then often a resampling is done. This is not really a necessary step in this pipeline. It's often done to have backward compatibility, so to say, downward compatibility with older results. Then a volume to surface mapping and a minimal smoothing. So before we extract and aggregate signals, we map the data from the volume. So the MRI scanner gives a volume, and we map it onto our reconstructed surface. And I'm talking a lot about surfaces and why this is important. Please bear with me. We'll come to that in a minute. and then a resampling to standard gray unit space and atlas spaces. So after we have processed the data, then we can aggregate it and average, for example, the signal over a brain region. And this is how this looks like with a GUI, for example. So this is how one operation looks like with a typical package. This is FSL. One loads basically the MRI image, the T1 image, and just hits run. And then one gets the original T1 weighted image and once the image where the skull was removed. And we can now nicely see this worked relatively nicely, except here in the front, we see the heuristic has failed. And this is something that will just happen very often. So this is basically all the results have always to be validated and checked. And so in addition to this problem, a problem is that there are many artifacts and distortions. So we don't need to go through them, but there is a long list of artifacts that can occur. And basically we need to check, is it there? Can be corrected? Often artifacts cannot be really corrected and the signal is just lost. So for example, even the static magnetic field, the B0 field, already produces an artifact because different substances have different susceptibilities for magnetization. Depending on whether there is a lot of blood or oxygen or whatever, the magnetic field lines will either be bent outwards or pushed inwards. And this leads to strong artifacts, as you can see here. So the signal is really lost, and this is the prefrontal cortex. We really want to have this, the prefrontal cortex, because it's very important. And here the signal is really compressed and stretched together. So what is often done is that, so the problem here is, this has to do with the phase encoding direction. So this is now very technical, but basically you can switch a certain parameter in an MRI scanner and can then get basically the artifact at another position. So we acquire the image once where the artifact is in the front, and there the sides and the back may be okay. And then we acquire the image again from the other side, and then another part may be okay. And as you can see here, these inhomogeneities really may delete a lot of information. So often half of the temporal cortex is just gone, or in the front is often a problem. This is another distortion. We can see it already here. So there is really this strong gradient going from left lower corner up to the upper right corner. And of course, if we would compare a signal here with a signal here, we would think, oh, this brain area is much darker than the area here. But this has nothing to do with metabolism or function or anything else. It's just an artifact. So we need to correct this if we really want to get meaningful results. And this artifact can then show up at various downstream operations as various glitches. Then, so for example, here the reconstructed surface is totally off. Here we see it completely misses the gray matter, and here also the white matter, while after it was corrected, it's still not perfect, but it's better at least. Eddy current induced distortion. So this is is also another kind of distortion induced by this eddy conduction. So this looks like this. These are just examples. So this is just to give you a hint how such artifacts may look like. So this is, for example, a movement induced artifact. So this would be the brain without motion, and here we can see there was some motion. So really, when you conduct such experiments, you really have to watch out for these things and really get a feeling, basically, for whether the signal and the data set is plausible or not. So you get this signal when you process a lot of when you really acquire this data, and you also put the subjects in the scanner, and you know the subjects, and then you ask them how does it feel, and they will maybe also you get also a hunch who may have moved a lot and so on. And it also depends a lot on how you prepare the subjects, how you put them in the scanner. So this is really a wide and complex field, and it has a lot to do with practical things, basically, that one only learns when one really tries to do such a processing. Yeah, so this is I will jump quickly over. This is just a correction, basically, how different images, this exemplifies the heuristic nature of MRI processing algorithms. So we have this blip up, blip down, and what I meant is different directions to acquire the image, and then basically one tries to find a correction for these images by combining these two images. So we perform, so this is the heuristic nature of these algorithms. We perform an initial guess on what could correct our artifact, and then we perform this correction, and then we look at the result, and if the result is good, we use it, and if not, we have to optimize further. And this is a very pervasive paradigm, basically, for correcting distortions and problems, basically. Yeah, we can see here left, we have a lot of movement. Right, the movement is corrected. And if you would use the data set on the left for your analysis, then you would never have a consistent signal. If you would average, for example, everything that is below my red laser pointer now, you would constantly average things from totally different brain regions and get totally different information. So these are all subtle things, but they are so numerous. Basically, one really has to go through every step and look after every step whether something like this happened or not. An important technique for MRI processing is image registration, and linear image registration is relatively okay. It doesn't do too much damage. So rigid body registration means that we really take the MRI volume and we just, what we maximally do, we rotate it in 3 directions and we shift it in 3 directions, maybe resize it globally. This is also relatively okay. So this is a registration technique that is relatively okay, and it's always needed. So we always need at least to register our different modalities, fMRI, T1, diffusion weighted, onto each other, such that we get, for example, the atlas from T1 onto diffusion weighted and fMRI. And registration is really a cool technique. Also, for example, so it's used through computer vision and imaging. So here you see images that have been taken from a rooftop in Munich heading towards the Alps. And usually you won't see the Alps, also not with your blank eyes or on this dizzy day. And then after taking several of these images and averaging them, one can suddenly see the Alps because the signal to noise ratio was improved, because we measured different photons at different times and got a better signal and could average the noise out of basically the noise that that diffracted the photons. And a very similar thing we can do also with MRI to increase signal to noise ratios. So here an example of an linear image registration. The gray is the diffusion weighted MRI, and the blue is the T1 weighted gray matter extracted from the T1. And as you can see here, they are in totally different spatial scales. So we first have to rescale and resize the T1 image such that it really matches, and that you can really use this to extract regional signals. And then a technique that is more problematic is nonlinear image registration, warping. So here we divide the brain into a grid. We lay a grid over the brain, and then we warp these grid lines basically onto each other. The problem here is really these algorithms are not able to identify and warp actual anatomical structures. They cannot identify anatomy or function or anything like this. They really just look for subtle intensity differences and use this for warping. And this is this of course deforms the whole brain. But this is a standard technique that was used like in many publications and for a long time, simply because nothing better was there. but now something better is there. So the problem for registration is that there is a high variability of cortical convolutions. So even if there are twins, so these are twins, A1 and A2, B1 and B2, C1 and C2, and if you compare their cortices, let's take a second and really compare the cortices of these twins. And you can see that even these twins have super different cortices. So you cannot even use twins basically to localize function or to registrate function based on their cortices. But yeah, so what we're doing here, so we're working now towards how can we localize function in the brain. And one important step is that we go from the volume space to the surface space. So left is our normal fMRI data set. It's a cube, it's an image cube, right? We have voxels, we have three-dimensional elements. Let's make life easier. And because the cortical surface is a sheet anyway, or it looks very sheety, we basically reconstruct the cortical surface and represent it as a two-dimensional surface. So here we can see this is now small triangles, basically. So these are small points, vertices, and edges that connect these vertices. And this is a representation of the folding pattern of the geometry of the cortical surface. And this contains now a much better information that is much more amendable for registration. Why is the surface better? So because of these convolutions, right? Because it is so convoluted. And if we now stretch this out on a sheet, so after we have this represented as this series of triangles, what we can do now is we can stretch this again out and look at it really as a two-dimensional object, because in the end it is then two-dimensional. We have lost this third dimension. This makes life easier. For example, because if we imagine, for example, if we would do an operation like smoothing or registration, then in the volume, the distance between two cortical regions in such a fold, such a sulcus, are very close, while in reality, these regions are actually wide apart. So along these lines, there's no connection between these regions. There's neither a white matter axon fire bundle, nor is there a local lateral connection. The connections run along this sheet or along the white matter. So it doesn't really make sense to work in volumes. The registration, so this is known now, has a severe problem. So if we really perform this non-linear segmentation registration, then we have a drift. There's a standard brain template that was created by registering many brains onto each other, and this brain template is actually now larger than average brains are, because there's a drift. They get larger and larger with every registration, and this is an obvious problem, but the not so obvious problem is that it doesn't actually identify brain structures. It's just really simple image registration and nonlinear warping of local intensity patterns. So the algorithm doesn't look for anything brainy. It just looks, is it dark there, or is it a bit brighter? Maybe it's the structure. This is all. And the advantage with the surface is that there we really only need to align structures within the 2D plane of the surface, while in a volume we also would have to shift around this plane of the surface. Okay, then yeah, even with surface-based registrations, there are still large distortions. I think we have only around half an hour left, I think. Yeah, I've many more slides, but it's no problem. It's the more important things can come at the beginning, and what comes at the end is less and less important for this lecture here. But it still may give an interesting outlook. But there won't come questions about it in the exam. So so we just saw the twins. There's a high intersubject variability, but even areas that are very well studied and yeah, well known, are highly variable, and no standard MRI brain atlas is able to capture this variation that was found with histology, basically, in post-mortem brains, basically. But this obviously cannot be used for living brains. And yeah, even the organization, the topology, the relative position of brain areas can be different, and even sometimes an area can be split and another area is in between. This happens only in a few people, but it can happen. And none of the standard techniques would be able to identify it. So basically this technique we are talking about here, the Human Connectome project, pipeline, surface reconstruction, and the glass atlas are the only ones that are able to deal with this. And this is, so now for comparison, this is how it was done previously. So there was extensive smoothing, and we probably know these images. So this sometimes people call this a bit mean blobology, because you have just these blobs. So this was very en vogue like 10, 20 years ago, and every magazine published these images, and there was the headline, brain region for religiosity, or for I don't know, for everything. A brain region was suddenly found, and this was done with these I would now call them old school techniques. Of course, these were valid at the time, and this was all meaningful. But if we now look at them, it's probably something we don't want to use anymore. A much better representation is now the gray ordinate space, where we basically combine the surface representation of the cortex together with a volume representation of subcortical structures. So subcortical structures, they are usually round, also so ball-like, so nuclei. For them, it's fine to use the volume representation, basically. And combining this basically with 90,000, around 90,000 gray ordinates, they are called here, one can get a nice subsampling of the brain. This is just an overview over the FreeSurfer Recon All steps, because FreeSurfer is such a widely used program, and this is a typical steps. So many things that we talked about now, again in a different visualization. So we have T1 weighted input, and then usually the skull is stripped or the brain extracted, then a volumetric labeling, which usually starts. So FreeSurfer has a good algorithms for identifying the gyral patterns, for example. So the gyros, central gyros and so on, they can be identified, but they may not be related to function. And then an intensity normalization, as we saw with all the kinds of artifacts that occur, then a white matter segmentation, then a surface atlas registration. So we register the surface, so we extract the surface, and then we pump it up to a ball, to a sphere, and then on the sphere, these patterns can be more easily aligned, basically. It's again this going from 3D to 2D. And why is the surface extraction coming after the? This looks strange. Okay. Anyway, yeah. So this is how this looks like when we basically register surfaces on a sphere. So they are pumped up, and then we see this gyros and sulcis, gyri and sulci here, and then we can register the gyri and sulci more easily here on the ball. So this is a standard step in FreeSurfer, but FreeSurfer is an older program. So now the HCP pipeline, the Human Connectome Project pipeline, uses this only just like a initial step. It doesn't really use the results, because the goal really is to localize function. We need to compare like with like. So if we have here a person and here a person, it turned out that it doesn't help us if we know where their precentral gyrus is, because there may not be the same function, but we really want to find the location where the populations encode or compute a certain function. This is our understanding. So we have brain areas and they compute function, basically. This is how we interpret this. So how can we localize function? As I described it, this was really done basically in the past in absence of better methods. What other options did we have than looking at the brain and, okay, this looks similar. Some of these results are still used today, like the Brodmann Atlas, which identified 47 areas based on the shape and organizations of neurons. So basically, we have, when we just look at the appearance, we only get a coarse understanding. So we need to have other modalities. We use multimodal data, multimodal features, instead of just folding patterns, for example. And relatively recently, algorithms were developed that can use myelin, resting state, functional connectivity, topographic maps, and receptor densities, and other things, basically everything, to every modality to align the cortex, the folded cortex. So myelin is this the sheets around neuron axons that make the signal travel faster, and it's nicely visible in MRI, basically. So here we have a map that shows myelin. So myelin is bright in T1 and dark in T2. So we don't see it directly when looking at T1 and T2 in isolation. But if we divide the T1 through the T2 image, of course, it must be in perfect registration. Otherwise, this doesn't work. So every voxel must perfectly overlap. Based noise or non-information reduces away, and we end up with information squared, so with an even better information that we are looking for. And when we then look at it, we can see that important structures are nicely visible in the myelin maps. So the sensory motor strip is super bright. The visual areas, the auditory areas, they can be very robustly localized using myelin maps. And when we compare registration based on myelin maps with registration based on folding on the sulci, then yeah, it just doesn't look as nice. So when the myelin registration is performed, then we see much sharper and much crisper structures. So this looks much smoother and crisper and more localized. And while with sulci, it looks more scattered and fragmented. Similar here, even if we use the resting state fMRI, so the people were just lying in a scanner doing nothing, and even if we just use the resting state fMRI signal to align their cortical surface on each other, we see then that even in task fMRI, the contrast is much much more focused, much more stronger if we compare pairs. So this is the task contrast, and we can see if this registration was done just based on folding patterns, then stuff diverges out. But when we add additional information, it gets much crisper and better in alignment, basically. So now that we can localize function, next step is to learn a subdivision in the brain based on function. And yeah, as we said, traditionally, researchers would just look at a single modality, and now we can compute such maps and gradients in many modalities, like function with fMRI, architecture from the T1 weighted images, connectivity using diffusion weighted MRI, and so on. And basically, we can now find a new definition of what constitutes an area. An area is where multiple of these measures are rapidly changing, because this indicates something is happening here. Here's the function rapidly changing, or the architecture or the connectivity is rapidly changing here. So this indicates that this could be a boundary of a region. So putting it all together, we say we want to identify this region here. So we look at an architectural pattern. So we see a strip of lightly myelinated cortex between the frontal eye field and premotor area. So it's not really visible here. This is the myelin map, but there's, yeah, the contrast doesn't allow it, but it's lightly myelinated, basically. This is the the gradient here. And then in the fMRI data, in the functional data, we see a unique task activity in the story task. So here's a strong contrast that is very closely located to this, what then finally will be the region. So the white dots basically show what will finally be the region. And we now look in different maps, how could we find this region basically? What constitutes this region? What are its characteristics? So we found an architectural characteristic. There's not much myelin. We found a unique contrast in a certain task. We found a certain connectivity pattern. We also know from the literature that the area largely corresponds to 55B. And with this, we can basically train a classifier that defines this region. If we now do this for all 150 to 200 cortical areas in each hemisphere. So this is a lot of work, but it was done. And it's also, with all of these multimodal features, they're all characterized and described for every of these almost 400 regions. And so we have for every, so what they do here basically is a binary classification. We have a relatively good, so we have a good cause understanding where the region is roughly located. So we have this large patch where the region will likely be. And then we train a classifier using all these features: myelin, thickness, task fMRI, resting state fMRI, isotopic maps, all kinds of things. Even artifacts can be used to train a classifier. Some regions are more susceptible to artifacts than others. So all of this can be used to train this classifier, and then it performs a binary classification to really segment out this region. So this is really a data-driven way to find function in different people. Yeah, let's jump over this. This is more context on how they found this. We have a few more other interesting things in the back. So I think we have now about 15 minutes to go. Maybe we also want to answer questions. So we have more like 10 minutes to go or so. So let's just highlight a few slides now that look interesting. So this is what comes out. So this is then the full multimodal parcellation, and you can see, so it looks really beautiful. I really love it. Then as an outlook, also microstructure can then integrate it again. So we have, for example, micrometer slices, and these can then again be used to augment this classification. So a thing about diffusion is that the actual fiber bundles are much smaller than the actual than the resolution of diffusion weighted MRI. And then let me just count this. Yeah, here it is. So the actual axons are small and the voxels are very large. So this is a big problem for diffusion weighted MRI. We cannot really measure the coupling strengths. Even if we could count like individual axons, there are still so many things like how many synaptic boutons are at the end, for example, which also have a huge impact on connectivity, or whether these axons target an excitatory and inhibitory population. So in a nutshell, all of this, what we get from the diffusion weighted MRI is really inferred. It's a model-based inference, basically. So this is, we cannot really identify weights. What we can is we can identify connection weights. We can't really identify connection weights. Sorry, let me start again. We cannot really identify connection weights. What we can identify is the strength of water diffusion in different directions. What we can then do is we can reweight in each voxel the strength of connection depending on the strength of diffusion. We can weight the strength of connection depending on the strength of diffusion in this direction, because in every voxel, fibers are crossing. And this is again a thing that we cannot really disentangle with diffusion weighted MRI. The fibers are, they can have all kinds of, here is the slide, have all kinds of orientation. They can be bent, kissing, crossing, fanning, and so on. And the diffusion signal would in all these situations look more or less the same. So this cannot be really distinguished in the diffusion signal, which is a problem. Another problem we talked about on the microscale, it's easy to parcel the brain. One node is one brain region, but how do we do it on a large scale? We saw how this is a problem. Quantifying edge strengths, yeah, we cannot really quantify the connection strength. Let me just turn off my camera because I'm going into another room because my son will be sleeping now. Just a second. Anyway, so there's a huge bias. There's a huge bias depending, for example, how many seeds you use to produce streamlines. So there are all kinds of biases that in the end can disrupt the success of the tractography, basically. Okay, so for the remaining minutes now, I would suggest to give you a quick overview about how these pipelines are installed in our supercomputing and cloud architecture. So eBrains basically is a platform for data and knowledge atlases, simulation services, medical data analytics. It offers all kinds of services for research. And these are all connected via a RESTful API and are interoperable. And researchers can use many different versions of these pipelines to compute results. Let me just show you a few highlights. So, for example, we have web GUIs where a project and brain simulation can be configured and started on a supercomputer. But also, of course, there's command line interfaces for that. Then a big problem with this is data privacy, basically. So this data is really very revealing. It tells us basically every voxel of an fMRI dataset is a fingerprint. And with that, we can reconstruct, for example, the fluid intelligence of the subject. And this was shown that this is possible basically to reconstruct the identity of subjects. So we need to protect these data. So we go over great lengths to protect this data. For example, with our image processing pipeline. So the image processing pipeline has three interfaces. One can just program it in shell, or one can use Jupyter notebooks, or one can start simulations on a supercomputer via such a GUI, a graphical user interface. And then basically on the supercomputer, an orchestrator and programs will ensure that the data is secured, for example, by encrypting it or by controlling access to these resources, or sandboxing, which means making the resources, shielding the resources from the rest of the system, or automation, which means that the process works largely autonomous and no human can actually come into contact with personal data. Yeah, for that we have a elaborate key exchange mechanism where secrets and keys are exchanged and shared in a secure manner, basically. And we are also building on pipelines. We are extending these pipelines and making them more generic and more customizable. All of this data also needs metadata in order to annotate it and to find it again. And then after all of this is done, then a computational result is often not reproducible, because to reproduce a computational result, really every last detail needs to be known, and there are so many details. So the actual article about the computational result is just advertising. The actual result is really everything, the whole environment, the whole software, everything that led to the production of the final figures. So to really get a reproducible result, or to get reproducible workflows, we need to version control data sets. We need to track the provenance of our digital objects. Where have they been changed, in which way, by whom, and when? And we need to track this really from the raw data to the final publication. We need to organize the data using standards and make them interoperable. We need to annotate them with metadata and documentation that makes them usable for others. Problems must be continuously solved, and with these continuous integration pipelines. Yeah, and for that, we use, for example, containerization, which is a great technique for bundling the whole environment together with the actual code. And data-led is what we use also. It's like Git for data. So GitHub and Git, you know, is version control for source code, for code files. And data-led basically extends this logic to arbitrary large files. And with this, we can really very specifically track which operations have been performed on a data set from the raw data to the final paper and the final figures. And we can prove that certain operations produced a certain result, which solves the reproducibility problem. So in summary, TVB uses access control, encryption, sandboxing, and automation to protect personal data in shared environments. The pipeline can be configured via web GUI, and containers capture all software and environment. And data captures all inputs, the containers, and how they were executed to produce the results. And with this, I'm now coming to the end. I think we have 5 minutes left, and I'm happy to take questions. Okay, thanks, Leon, for